Dear students, welcome to my new channel. Today we will discuss on the topic of biopharmaceutical considerations in dosage form design under the chapter of gastrointestinal drug absorption. So let's start. So as we have seen, drug in tablet or granules form or in small particle form will ultimately be in solution from where it will get absorbed. So biopharmaceutical factors in dosage form design or factors influencing the GI absorption of drug includes some pharmaceutical factors and patient-related factors. So we'll begin with pharmaceutical factors. So pharmaceuticals factor can be described in two different categories like physicochemical properties of drug and other factors related to dosage form or dosage form related or pharmaceutical excipient related. These are called pharmacotechnical factor. So let's start with physicochemical properties. So the physicochemical properties of drug include drug solubility and dissolution rate. The more dissolution would happen to the drug, the more absorption would we have. Then particle size and effective surface area. The more smaller the particle size, the more would be the absorption or dissolution. Then polymorphism or amorphism. This is the different crystal structure of any drug substance. So they have their effect on ADME of the drug. Then pseudopolymorphism, that is presence of hydrates or solvents into the structure of the drug. Salt form of the drug, this will ultimately have better solubility. Lipophilicity of the drug, the more lipophilic the drug, the more would be the absorption. pK of the drug and gastrointestinal pH. So the difference between the pK and pH will ultimately give rise to ionization or unionized form of the drug, which will ultimately affect the absorption of the drug. Then drug stability, and lastly stereochemical nature of the drug. So the pharmacotechnical factors include disintegration time. The more disintegration time would a drug have, then its disintegration will be decreased. Okay, this will ultimately influence the dissolution time. The manufacturing variables like compression, drying, mixing rate, the pharmaceutical ingredients, nature and type of dosage form, product age and storage condition. So let's start with physicochemical properties. First of all, particle size and effective surface area. As we have already said that reducing the particle size will enhance the surface area, and this will ultimately give rise to better absorption. So solubility, the phenomenon of dissolution of solute in a solvent to give a homogeneous system. The process by which a solid or liquid forms a homogeneous mixture with a solvent or mass transfer from a solid to liquid phase. Particle size and surface area of a solid drug are inversely related to each other. Smaller the drug particle, greater the surface area, as we have already discussed. So there are two surface area. One is absolute and another is effective. We'll discuss this later. So as we are talking about the particle size, so micronization has in fact enabled the formulator to decrease the dose of certain drugs because of increased absorption. So this is the reduction of the particle size. Grasophylline dose was reduced to half and that of spironolactone was decreased to 20 times following micronization, and their bioavailability was enhanced. For lipophilic drug, aspirin, phenacetin, or phenobarbital, micronization actually results in decrease in the effective surface area. Why? Because these are lipophilic drug. So these will ultimately give rise to more lipophilic surface, and the more lipophilic a drug would be, then the more would be decrease in its solubility. Okay, so it leads to fall in dissolution rate. So for the better understanding of surface area, the absolute or effective surface area, we can focus on these figures. For example, absolute surface area. So what do you mean by absolute? Absolute means total. For example, we sometimes say that you are absolutely right. That is, you are totally right. Okay. So the red and blue marks are both absolute surface area. That is, the total surface will count for this absolute surface area, the blue ones or the red ones. Okay, but if we talk about the effective surface area, then surface area exposed to the dissolution medium would be the effective surface area. This surface, the red surface, would be the effective surface area because these are actually in contact with the dissolution media, that is water. Okay, but water cannot easily get entered into the this surface area. So you know, effective surface area will ultimately result in the overall dissolution of the drug. So in conclusion, effective surface area is important. Greater the effective surface area, greater the contact between the solute and solvent, and greater the solubility, dissolution, adsorption, and bioavailability. However, absolute surface area of any hydrophobic drug can be converted into effective surface area by several methods. For example, use of surfactants decrease the interfacial tension and thus allows the solvent to enter into groups. It also displaces the absorbed air with solvent. For example, 280 promotes the wettability and thus increase the bioavailability of phenacetin. Okay, if we use surfactant, then this will ultimately reduce the surface tension and will ultimately enhance the better contact of water with the surfaces. Okay, the next one is addition of hydrophilic diluent, for example polyethylene glycol, polyvinyl pyrrolidone, dextrose, that coat the surface of the hydrophobic drug particle and render them hydrophilic. So we can see here the micronation. If we reduce the size, then this will ultimately give rise to a better surface area. Okay, so size reduction is also called micronization. Suggested to go for micronization of API for better solubility. Go for micronization up to 0.1 micron only. Extreme micronization develop a charge on particle, so be careful about the micronization. This charge will interfere the adsorption, not advisable for unstable drug like penicillin, erythromycin, because they cause undesirable side effect like gastric irritation. So we can have a better uh, overview of the impact of particle size reduction. For example, if you see these are the fine particles, the greens are the medium sized particles, and the below one are the coarse particle. So you can see the coarse particle dissolution was less. Okay, if the more the surface area, the more blood concentration will ultimately have. Okay, and in case of fine and use of surfactant, we can see this has ultimately increased the dissolution in a greater way. So micronization is not worked out in hydrophobic drug, as we have already discussed. So hydrophobic drug has absorbed air. Particles reaggregate to form bigger particles. Surface may get charged due to poor permeation. So our next physicochemical factor is polymorphism. Substance exists in more than one crystalline form is called polymorph, and this phenomenon is called polymorphism. For example, 65% of sulfonamides exhibit polymorphism. For example, phenobarbital, methylparaben, sulfapyridine exist in more than 6 forms. Cortisone acetate exists in 8 forms. So you can see uh, this is the different polymorphs of a same drug. Okay. This is showing one arrangement, this polymorph is showing another arrangement. Okay, but these three are of same active substances but having different shape. We can have a better idea about polymorphism by focusing on these figures. In this figure we can see diamond, these are crystal shape. Okay, but this is also a carbon, this is also a carbon, but this is a coal which is amorphous, not having a particular shape or structure. So structure makes big difference, we can easily understand. So these are two polymorph of carbon. Same, we can say that chloramphenicol permeate are available in three polymorphs A, B, C. Among these three, A is biologically inactive, but B is best absorbed from the GI tract. So we use usually B polymorph. Okay. Uh, if we categorize polymorphism, we would find out that So internal structure of a compound can be of two types, that is crystalline. We can see that crystal structure is very much organized, but we can have another one that is non-crystalline. These are also called amorphous, okay, because no particular structure is seen here. However, we'll study about these crystalline shapes. This can be divided into two forms. One is polymorph and another is molecular adducts. So polymorph are anisotropic or monotropic, and molecular adducts, these are non-stoichiometric, stoichiometric and stoichiometric complexes. And in case of stoichiometric complexes, these are also called pseudopolymorphs. Okay, this can be of two types: organic solvents and hydrates. We'll discuss all of this later. So polymorph vary in their physical properties like solubility, melting point, density, hardness, compressibility, stability. Crystallizations, process of preparation of crystals from solvent. Analysis of crystal can be done in optical crystallography, X-ray diffraction, differential scanning calorimeter, DSC. So we can see here that a drug can be in stable or unstable form or metastable form. However, whenever a drug is having more stability, then it would not prefer to go in dissolution, to go in absorption, metabolism, excretion, because its condition is static. Okay, so the more stable a drug, the lower energy state it would have, then it would ultimately have a highest melting point. That is, that's why it is more stable. That is, it would not try to react or try to attend in any of the reaction. That's why it are having less disintegration, less dissolution, less absorption, less viability, less the solubility. Okay, these are the reasons why stable polymorph will not have a better dissolution. But in case of metastable, these are just opposite to our stable form. Highest energy state, lowest melting point, more aqueous solubility, more disintegration, more dissolution, more absorption, more viability. Okay. So as we have shown earlier that there is difference between crystal and amorphous. Crystalline shape is some particular shape. Okay, this is a crystal shape, but amorphous shape don't have a particular structure. So novobiocene crystalline form, novobiocene amorphous form. So what would be the difference? So crystal shape is stable one. Okay, but amorphous phase don't are not stable. So as crystal shape are stable, so these have lowest energy state. These are more stable. As these are more stable, they will have more melting point, and uh, unfortunately, these will ultimately have less solution, less dissolution, less adsorption, less bioavailability, and these are preferred for sustained release formulation. So in case of amorphous phase, the conditions would be just opposite to crystal shape. So stable, metastable polymorph we have already discussed. If we focus on the graph, we can find out that A is the stable polymorph. Here is the A, the lower one, but stable polymorph will not participate, will hardly participate in dissolution or adsorption. That's why their blood concentration is lower. In case of B, we can see that if we use 100% B, that is Ba is having that is the metastable is having the more soluble form because of uh, its lower melting point and higher aqueous solubilities, and C is the unstable polymorph which would be between these two. So the amorphous form, these are having no internal crystalline structure. Higher energy state can be considered as supercooled liquid. They have greater aqueous solubility than the crystalline form. So amorphous form of novobiocene is 10 times more soluble than the crystalline form. Chloramphenicol palmitate, cortisone acetate, phenobarbital are other examples where the amorphous forms exhibit higher water solubility. So that's why we tend to use the amorphous form for better solubility. We can see here the crystalline solid. This is the amorphous solid. The crystalline is stable. there is less internal energy. But in case of amorphous solid, the particular arrangement is not uniform. That's why these are having more dynamic condition within its structure. That's why dissolution is a dynamic condition. So these will ultimately go to dissolution in a better way. So amorphous solid has higher energy than its corresponding crystalline solid, therefore higher apparent solubility and dissolution rate. So you can see here the amorphous have higher solubility and the crystalline are having lower solubility. So our next one is stoichiometric complexes. Crystals are made from solvents only. During crystallization, there is a chance of entrapment of solvent molecules in crystal. Those crystals are called stoichiometric complexes. You can see here it can be of two types: hydrates and organic solvents. Hydrates means water, and organic solvents, these are less common, but hydrates are more common. So we'll discuss this. So a crystal, a stoichiometric form can have hydrates or anhydrate form. So anhydrate means less absence of water. Okay, and hydrate means presence of water in the molecule or in the structure. So as these are already having less water or no water, they have greater chance to absorb more water. These are like dry soil. Okay, if you see the dry silica or any dry soil will absorb water more, but in case of hydrate, these will not absorb as that one. So hydrates are less soluble, less dissolution, less absorption, less viability, but anhydrates have more solubility, more dissolution, more viability. So hydrates are already in interaction with water, as we have already discussed. They have already water, so they're thermodynamically in higher state. So higher state means less energy for crystal breakup. So we have already understand that what would be the order of dissolution. Anhydrate would have the highest dissolution, monohydrate would have the next highest dissolution, and trihydrate will have the lowest dissolution, because these have already the water content in its structure. So we know the dissolution process of a salt form of weakly acidic drug in distilled fluid. Um, Sometimes we give the drug in salt form, for example, diclofenac sodium, diclofenac potassium. Okay, so there are some reasons for this. We can say if we give any drug in a salt form, its solubility will be enhanced. Okay, and if our salt form, the size of the counter ion, that is the less size of the counter ion, the more solubility should have. Okay, smaller the size of the counter ion, greater the solubility. That's why a drug having uh, a potassium salt and calcium salt, ultimately in comparison with the free acid, these both salt will have better dissolution, but between these two, potassium would have the better solution. Why? Because potassium is having the lower molecular structure, that is lower size of its structure. So these are the overall scenario you can already understand from our discussion. Our next important topic is pH partition hypothesis. This is the most important concept to be understood in this lecture. So according to pH partition hypothesis, the GI epithelia acts as a lipid barrier toward drugs which are absorbed by passive diffusion, and those that are lipid soluble will pass across the barrier. The theory states that for drug compounds of molecular weight greater than 100, which are primarily transported across the biomembrane by passive diffusion, the process of absorption is governed by dissociation constant of the drug, lipid solubility of the ionized drug, and the pH of the absorption sites. So we'll discuss this broadly later. Most important thing that we need to understand is that the GI tract is a simple lipoidal barrier to the transport of drug. Larger the fraction of the ionized drug, faster the absorption. Greater the lipophilicity of the ionized drug, better the absorption. So for better absorption, uh, the drug need to be very unionized and undissociated. Okay. So better the lipophilicity of the drug, better the absorption. So this is the main aspect of phase partition theory. We have already discussed in our previous lecture the Henderson-Hasselbalch equation. It is the lower the peak of any drug, the stronger the acid. the greater the portion of the ionized form of the particular pH. The higher the peak of the basic drug, stronger the basic, the greater the proportion of ionized form of the particular pH. So we'll discuss this later. However, there are some limitations of the pH partition hypothesis because presence of virtual membrane absorption of the ionized drug, because pH partition theory always described with the unionized drug. But what about the absorption of ionized drug? Influence of GI surface area and residence time of the drug, presence of aqueous unstirred diffusion layer. We can say here the unstirred diffusion layer, absorption of the ionized drug, and influence of the GI surface area and residence time of the drug. These are not described in this hypothesis. So now, according to the pH partition hypothesis, we have seen that drugs solubility and absorption better depends on the pH of the GI tract and pK of the drug. Okay, so according to the Henderson-Hasselbalch equation, we know the pH is equal to pK plus log of salt by acid. We already learned this in our previous lectures. So what this equation says, actually, log of salt by acid, the salt is will be in ionized form. Okay, why salt is ionized form? For example, sodium chloride remains in the solution as ionized, like sodium plus and chloride minus. So salt form will always be ionized, and acid form will not be ionized. For example, if we talk about an acid, for example CH3COOH, there is no ion here. Okay, but in the salt form we can see ions like plus, minus. So from now on we need to learn that acid or base will have the unionized form, and salt will have ionized form. So let's discuss. So the equation, Henderson-Hasselbalch equation, can be written in that way after derivation. The salt by acid would be equal to 10 to the power pH minus pK. In case of base, base by salt would be equal to this. Here the salt form is ionized form, and acid or base form is unionized. So these will ultimately help in better understanding of the dissolution or the absorption of the drug. So we need to learn very carefully this. We can see the pK value of weak acids and weak bases. Uh, Here we can say that stronger acid. So we know stronger acid. Just think about stronger acid and stronger base. Just think about like HCl or NaOH, sodium hydroxide and HCl. They will be completely ionized throughout the all pH value. Okay, I'm talking this in case of solvents like HCl and sodium hydroxide. So stronger acid and bases would always be ionized in all pH value. So as we have already discussed, ionized form will have poorly absorbed from the GI tract. So ionized form will have poor absorption from the GI tract. So we have already understood these two. Okay. Now move on the next phenomenon, that is very weak acid and very weak base. What would ultimately happen to them? That is very weak acid and very weak base. So we'll discuss about these two. So please keep in mind that very weak acid will always be unionized, and very weak base will also be unionized, because they will not very weak acid will not release the proton in the GI fluid. Okay, because they are very weak. They are not as stronger as like a strong acid, because strong acid will release protein very promptly, but very weak acid cannot do that. That's why as they are not releasing any proton, that is the charged, uh, this will ultimately result in no charge or no ion in the solution. That's why they will ionized at all pH value, because they are very weak. This is as like as the very weak bases, because they will never release in a hydroxyl group, that is negative ion. Okay, so they will always be unionized at all pH value. So the most important thing is we need to learn about moderately weak acid and moderately weak bases. These two are very important because most of our drugs are either moderately weak acid or moderately weak base. Okay, so what is happening? Let's just think that a weak acid, if it is in stomach, Okay, it would be in contact with HCl, that is acidic condition. So acid in acid, a weak acid in acid will be unionized. Okay, why unionized? Because if acid come in contact with base, then it will form salt. Okay, and salt is ionized form. In case of weak acid, whenever it is in gastric pH, it will remain unionized because it will not form any salt. Okay, but if this weak acid travels into the intestine, what will happen? It will be in contact with base. Okay, because intestinal pH is basic. So acid plus base, what will ultimately result? It will ultimately result in salt formation, and we know salt is ionized. That's why the ionized portion will never ever be uh, easily absorbed from the intestine. That's why weakly acidic drug will better absorb in where? It will be better absorbed in GI tract, that is stomach. And this is like as the weakly base. Weakly base will ionized at gastric pH and ionized at intestinal pH. So you may have confusion regarding this. This confusion will be solved in our next lecture. We'll discuss about the problem and solution. So let's see here. A weakly acidic drug has a pK of 3.5. Very good. So now, if the pH of the stomach is 2.1, then we have to calculate the ratio of ionized and unionized using the equation. So now tell me, what would be the fate of a weakly acidic drug in stomach? What would happen? As a theory, we know that weakly acidic drug will be unionized in the stomach and will be better absorbed from stomach. If you see the question 2, then what would we can see that the drug has already passed the stomach and come into the small intestine where the pH is 6. We'll see what would happen in this intestinal area after drug comes into the intestine. Okay, as a theory, we can already understand that in the intestine, the drug would be in, as the drug is acid and the intestine is base. What would ultimately happen? This will ultimately result in salt, and we know the drug would be in salt form in the where? In the intestine. So salt form means ionized form. The drug would be highly ionized, and you know, if any drug is in ionized form, what would happen? This will decrease the bioavailability or the absorption of the drug. Okay, so now let's see the uh, problem, the solution of the problem. Let's see. So here is the ultimate solution of the math that have already been given earlier. We can see after the derivation, after the calculation, we have seen that the salt and acid ratio is 0.03. Means the acidic portion is higher, that is the unionized portion is higher in the stomach. Okay, and this is the solve of the question number 2. We can see the salt and acid ratio was what was 316. That means in the intestine, in the small intestine, drug is the salt concentration is higher and ionized form is higher, and drug would not be absorbed from this intestine. Drug would eventually absorbed in the stomach acid. Okay, I think you have already understood this. Anyway, shortcut formula for question 1 and 2 we have already shown in our previous lecture, that this is the derivation of our Henderson-Hasselbalch equation. If you just calculate, if you just put the value of pH and pK in this equation, we will ultimately get the result. So that means salt by acid is equal to 0.03. This is the result of our first problem. So better absorbed in stomach. And in case of next equation or next problem, we saw that salt by acid ratio was 316. That means salt is higher in concentration, that is more ionized in intestine, that is less absorption would happen. So we have another practice problem for weakly basic drug that has a pK of 9.45. And this is the pK of propranolol. Anyway, so in the stomach, what would ultimately happen? And in the small intestine, what would happen? In the stomach, as it is basic drug, the stomach condition is acidic, it will not tend to form salt here. Sorry, it will not tend to form salt in small intestine, but in stomach, what will ultimately happen? It will form salt. Okay, because stomach is acidic, this is basic, it will form salt, and the salt is ionized, and it will not absorb from where? From stomach. But in small intestine, as theory says, pK and pH value, this will have some more absorption from the small intestine. If you see the result, we'll find that um, from the stomach, as we have discussed, very poor absorption. But in small intestine, these are having better absorption than stomach. Okay, because 10 to the power minus 9, very low, and it's million time faster absorption in small intestine. So now let's dive into our pharmaceutical factors like disintegration time, dissolution time, manufacturing variables, pharmaceutical ingredients, nature and type of dosage form, product age and storage condition. So the disintegration time. So we have already discussed coated tablets, especially sugar coated, have longer disintegration time. Disintegration time is directly proportional to the amount of the binder present in the compressional force of the tablet. Then the manufacturing process variables. So method of granulation and compression force ultimately affect the dissolution. We can see as a rule of thumb, if we compress a tablet very promptly or very strongly, what would happen? The compressed tablet would have a higher binding affinity, that is the tablet would be very compact and very hard. So ultimately dissolution will decreased. But in some of the cases, dissolution increased for some of the excipients and drugs. What happened to them? In case of those drugs, what the granule shape is like that. But whenever we are compressing in a very robust way, what is ultimately happening? The granule shape would be like that. Because if you compress a granule, its shape would be like that, like a disc. Okay, whenever it is forming a disc shape, what will ultimately happen? This will ultimately increase the surface area. Okay, and this increased surface area will ultimately raise the dissolution of the tablet. So in that case, for some of the materials, dissolution has increased. Okay, and for some of the drugs, two of them can happen. This has been shown in CMD. So pharmaceutical ingredients. So excipients used to ensure the acceptability of physicochemical stability during the shelf life, uniformity and composition and doses, and optimum viability and functionality of the drug product. Nature and type of dosage form, we know solution will have the highest dissolution, then emulsion, then suspension. These are all liquid dosage form. Then capsule, then tablet, then coated tablet, because these are coated. There would be a duration or lag time for release of the coated drug, then enteric coated, and lastly the sustained release product. So these are the some patient related factors regarding uh, our biopharmaceutical absorption of the drug in dosage form. So we'll discuss this one by one. Gastric emptying. uh, Factors affecting the gastric emptying includes volume of the ingested material, then fatty and carbohydrate food will decrease the gastric absorption. Temperature of the food will increase the emptying rate, because any addition of temperature will increase any of the reaction. Then body position. Uh, if we lie on the left side, it will decrease emptying rate because of the position. Then some anticholinergic, narcotic, and analgesic drug will ultimately decrease absorption. So there are other factors influencing the absorption, that is viscosity, then emotional state, because stressful condition will increase the stomach contraction, but in case of depression, the neurotransmitter release would be decreased, and this will ultimately decrease the uh, emptying. Then disease state. Some diabetic patient, hypothyroidism, rate of emptying is decreased in hyperthyroidism. For exercise, it reduces the emptying rate because in case of exercise, what happened? Our blood circulation uh, get very high in the muscle area, but this ultimately reduce the blood circulation in the GI tract. That's why this reduce the gastric emptying. So these are all from the biopharmaceutical factors affecting the dosage form. If you have any confusion or question, please inform me in the comment section. Thank you.